Thanks again for the invitation. It's great to be here. I've learned a lot at this meeting, and so it's been great so far. So I wanted to today talk a little bit about some recent projects in looking at exosomal RNA payloads and using them to power next generation early cancer detection and residual minimal disease detection biomarkers in prostate and liver cancer. And I hope to show you some aspects of the data analysis that we carried out and give you a flavor of their potential promise in the coming years in medicine. So this is, of course, joint work. I won't have time to cover everything, so you can look at the details in this paper. Both of them are in the preprints. Here's one on the direct subject matter of today's talk, and here's another one that's very much related to that. The work in liver cancer was led at Mount Sinai, but IBM, under the direction of Gustavo Stolovitzky, led the work in prostate cancer. Okay, so the outline is basically, I'll give you the study design, the rationale for looking at hepatocellular carcinoma, the most frequent form of liver cancer. We'll spend the most time on the data-driven analytic approach that we've developed for genomic target identification in exosomal RNA. And that'll give you a little flavor of how well it performs in a independent clinical validation study. And hopefully we'll have time for questions both now and in the panel. Okay, so just really briefly, HCC is one of the most common tumors in the world. It's the third leading cause of cancer deaths worldwide. Hepatitis B is a major etiological factor, but the key feature of HCC is that it almost always occurs in the background of significant end-stage liver disease, such as cirrhosis. So there's this long latency time of significant disease, which is really bad for when it rarely, but more and more frequently, and in recent years, develops into HCC because your treatment options are quite limited with a very severely diseased liver. So the need for liquid biopsies is huge in this disease because often early detection is actually itself a cure. So just briefly, we recently surveyed the liquid biopsy space of HCC, including EV-based studies, cfDNA, and so on, in case you're interested in learning more about this from a more global perspective. So in our particular study, we leveraged two feature discovery sets across two cancer data sets, so basically prostate and liver cancer. And then we had one large external validation data set in liver cancer exclusively. So the prostate cancer data set was distinguished by having EVs from multiple biofluids, in this case urine and serum, and multiple isolation techniques, including ultracentrifugation and more non-invasive lab-on-a-chip technologies that Gustavo Stolovitzky's group at IBM have pioneered. And finally, also to look at the small RNA by kind of locus of origin, and looking at those specifically enriched from EVs and those that come from cellular RNA. So the isolation of EVs and sequencing of small RNA was carried out across matched patient urine and serum biofluids. And like I mentioned, EV isolation was carried out through classic ultracentrifugation and this nano-DLD. So we carried out a detailed study characterizing small RNA expression profiles across these three major axes of variation, and came up with a list of properties that allowed us to bucket these small RNAs into various groups of interest. Using this feature set, and these properties, we then turned to profiling a small liver cancer case control discovery set of 15 patients to discover ultimately 3 promising biomarker candidates, which we then moved on to validate the performance of an independent phase 2 clinical study. So really the main outcome of these informatics studies was that that essentially these properties, these informatic properties, would allow us to sensitively characterize EV-specific small RNA clusters. And what we found was that they emanated largely from unannotated loci in the genome. So this prompted us to take a different approach to analyzing our data. And it's worth taking a closer look at the biological rationale for this. Essentially, EVs, as you know, are enriched in small RNA, which immediately gives you a multiple mapping problem as a bioinformatic consequence. Further, essentially as well, since small RNAs don't have obvious sequence features such as open reading frames, splicing motifs, and other features, they're generally a little more difficult to broadly characterize. We do have some clues, though, on how to characterize them, since almost all small RNAs derive from post-transcriptional processing of larger RNA precursors. So we know that some small RNA come from hairpins. They yield a single functional RNA species after processing, but that isn't true generally. Many other types of small RNAs, small RNA genes produce a really diverse population of mature small RNAs from a single, often inferred, larger precursor. So that left us with the notion that we could take a very empirical definition and essentially rely on a strand-sensitive alignment pattern notion of a small RNA gene and characterize those in a very data-driven fashion. So that was essentially our approach. And what we came up with was looking at all the alignments of the small RNA reads to genomic reference. We defined clusters of contiguous genomic regions with essentially sufficient alignment coverage, which is just a threshold that we set. And we called these contiguous regions small RNA clusters, or as we lovingly called them, SMERCs. So this allowed us to capture the known heterogeneous genome-wide expression of clusters of these small RNA precursors, each of which, again, can give rise to multiple functional small RNA products. So adjacent SMERCs were merged if they overlapped within a minimum padding threshold. And we essentially used this moving average window approach to look at these clusters. And we came up with a couple of defining properties to delineate them. So the first was entropy, essentially a read tiling efficiency or complexity, which allowed us to look at the ratio of unique read nucleotide sequences to total coverage. The next was peak coverage, looking at the total number of reads and the ratio of reads that were contained in the small RNA cluster peak to the total coverage. And finally, we looked at the consensus sequence of each small RNA cluster to help us understand binding properties to RNA binding proteins and other details. So the set of all of these small RNA clusters allowed us to essentially compute the accumulation loci of small RNA expression, some of which were comprised of microRNA, but many others which were not. And what we found was that the statistical properties of these small RNA clusters were quite similar to regular RNA sequencing. The standard over-dispersed negative binomial distribution applied, and so on. And what we were able to do then was study the major variations of expression across the axes that I mentioned. So across EV isolation technique, we recovered the kind of expected high false positive rate of UC for lowly expressed small RNA clusters compared to the non-violent EV isolation technique, nano DLT, and we had a very good correspondence for higher expression. We also found a bimodal, essentially a bimodal distribution of small RNA clusters in peak coverage and small RNA cluster entropy, which allowed us to cleanly separate cellular and EV expressed small RNA clusters. So taken together, this whole exercise really amounted to developing a very simple-minded first principles method for characterizing and potentially ranking in a very data-driven, assumption-free manner the exosomal RNA payloads. And one of the key upshots is that the majority of these loci really emanate from unannotated genomic loci. So we use this technique to look at our ERCC exosomal RNA discovery set. We found 3 unannotated exosomal RNA biomarkers. And we proceeded from that point to investigate. But just as a brief mention, I can basically show you that the small RNA clusters very cleanly distinguish samples from each other and sample groups from each other. Here you can see serum EVs very cleanly separating out from urine EVs profiles, and then both groups very nicely separating out from the RNA payloads that aren't necessarily packed into exosomes. So finally, the upshot is really what is the outcome of this investigation in terms of human health. We tested our 3 exosomally specific RNA unannotated biomarkers in a phase 2 independent clinical validation study. Here I've plotted the ROC curve that shows how well our 3 biomarkers do in a logistic regression model for predicting risk of early cancer incidence among a population of severe liver cancer patients. Here is the clinical standard of using a biomarker that's been around since the 70s plus ultrasound. And the model that we've come up with just using the exosomal payloads is in red. When we combine our model with the kind of canonical biomarker that people have been using since the 70s, it actually improves it considerably. And so this really raises hopes that we could use some of these results to create an operator-independent, completely blood-based early detection panel for liver cancer. So so we're coming up to the end here, but I hope that I've described some of the salient features of our data-driven exosomal RNA payload discovery technique. Essentially, here's the graphical study summary, but the conclusion is, from my perspective and from data analysis perspective, that there's an enormous amount of genomic small RNA dark matter out there, and that relying on present annotations, which, even though they're improving, may actually allow us to miss quite a bit of low-hanging fruit, which, even though it may not be functionally characterized, could be very important in the short term for liquid biopsies and other applications. So thanks a lot. I'll stop here and take any questions.
Thanks, Bojan. That is very interesting. We've got a couple minutes for questions, and someone asks, do you think the unannotated RNA are uncharacterized microRNA?
Yeah, that's a great question. So typically these uncharacterized RNA are not in the size range of microRNA. They're a little smaller. And what we found is that there's evidence that they're actually smaller than the microRNA and in complex with several key classes of RNA binding proteins, including splicing complexes. So there are certainly some microRNA, but that's more an exception, actually.
I'll ask the open-ended question, what do you think they are?
Well, we don't know functionally what they do. Since we don't have lots of annotation data for them, we don't even know really what they are. So, it's going to be a slow slog through functional assays to really figure out the real answer. But it is encouraging that a lot of them share common RNA binding protein motifs.
Can you say that last sentence again? They do or they do not?
They do. So even though they might be distinct and uncharacterized, it seems like large classes of them bind or preferentially bind well to only several RNA binding proteins.
And I apologize if you addressed this already, but did you look at where in the transcriptome, where in the genome these unannotated clusters are? Is there some commonality?
Yeah, so they can occur in genomic deserts. Some of the ones that, in fact, 2 of the ones that we had for our biomarker panel result from intronic sequence of relatively obscure genes, if you will. So they really show no preference in localization.
Here's a question. I'm not sure I understand it. What percentage of unannotated exoRNAs you had in your samples?
Yeah, that's a great question. So the overwhelming majority are unannotated. Let me see if I can pull up this figure right here. So if you look at the middle pane, it's right here. This bump right here is the percentage of fully overlapping features that we found from these small RNA clusters. And this is the dominant mode, which is very, very low to 0 overlap right here. So you can see that of the ones that overlap, the majority are microRNA, we have SNO-RNA and so on and so forth.
So basically what you're telling us is we already don't know what most of the annotated non-coding RNAs are doing in our biofluids, and you've just discovered an even more mysterious class of...
Well, what we've discovered is that the vast majority of even actionable signal for in the context of this liquid biopsy comes from the unannotated, completely unknown dark matter, if you will.
All right, well, thanks so much.
Can I
Oh,
have the last questions?
sure.
Why do you select that 3 unannotated features? And how about other classes?
Yeah, that's a great question. So we selected them for pragmatic reasons as well as informatic ones. So I mentioned the 3 criteria that we had before, which allowed us to better select exosomal versus cellular and various other properties that allow us to kind of try and rank them. But the second was more pragmatic because we ultimately had to design qPCR primers for validating in the clinical study. And so for those small RNA that were close to low complexity sequence and had other obstacles for a clean signal, we favored ones that did not have those problems. So it was a mix of pragmatic and informatic considerations.
Can you explain a little bit clear? So why among all the annotated features did you identify biomarker there or not?
So we identified the features that had the strongest segregating power between cases and controls. And unless they were difficult to amplify and unless they were difficult to design primers for, we retained the top 3.
Okay, so by chance that the 3 are all being annotated.
Well, the chance is actually rather good, just given the distribution in the background that I just showed. So this is the, if you will, the underlying probability distribution that we'd find something that actually was fully annotated. That's this mode. Here you see the majority of the data are not very well captured by present annotations.
Okay, got it.